In this picture, we show how fatty acid metabolism fits in the grand scheme of metabolism and how it interacts with the other pathways of metabolism. In this picture, we show how fatty acid metabolism fits in the grand scheme of metabolism and how it interacts with the other pathways of metabolism. What are fatty acids? Fatty acids consist of a hydrocarbon acyl chain that consists simply of carbon and hydrogen molecules, and with a carboxylic acid group at one end. Some fatty acids are unsaturated, that is, they have double bonds in their acid chains. Now, this is a molecule of oleate, which is an example of that. Now, we use a nomenclature to describe fatty acids. Palmitate, which is a saturated fatty acid, has 16 carbons, so the first number there. We follow that with a colon mark, and then a 0 indicating that it contains no double bonds. Oleate, on the other hand, we describe as 18 because it has 18 carbons, colon mark, and 1, which stands for 1 double bond. Now, some of the more common fatty acids that we may encounter are lauric acid, which is 12 carbons, no double bonds. Myristic acid, which is 14 0. Palmitic acid, probably the most common fatty acid, which is 16 0. Stearic acid, which is 18 0. And then the unsaturated fatty acids, which include oleic acid, 18 1, linoleic acid, 18 2, linolenic acid, 18 3, and arachidonic acid, 24. There are other fatty acids, but these are the most common ones that you'll encounter. Fatty acids are synthesized from acetyl-CoA in a process that requires both NADPH and ATP. And the total reaction is indicated below. 8 acetyl-CoAs plus 14 NADPHs plus 7 ATPs produces a molecule of palmitate, 16-0, plus 14 NADP pluses plus 7 ADPs and 8 molecules of CoA. Now, fatty acid synthesis occurs in the cytosol. So acetyl-CoA, which is generated by pyruvate dehydrogenase in the mitochondria, has to be transported out of the mitochondria in order to be converted to fatty acids. And this occurs in a process called the citrate shuttle. So you have a molecule of pyruvate, which is converted using ATP to oxaloacetate. Oxaloacetate combines with acetyl-CoA to form citrate. Now, you recognize this probably as the first step in the TCA cycle. But rather than the citrate continuing in the TCA cycle, in this particular case, it is shuttled outside of the mitochondria into the cytosol. There, the citrate is converted back into oxaloacetate, again consuming a molecule of ATP and generating the molecule of acetyl-CoA. Oxaloacetate is converted to malate, consuming a molecule of NADH, and malate is converted to pyruvate, consuming a molecule of CO2, and producing a molecule of NADPH in the process. The pyruvate then is shuttled back into the mitochondria, and the process can be repeated. Now note that the citrate shuttle produces 1 cytosolic NADPH. So in the transport of 8 acetyl-CoAs, 8 molecules of NADPH are produced. This leaves 6 additional NADPHs which are required for palmitate synthesis, and these are produced by the pentose phosphate shunt. The rate-limiting and regulated step in fatty acid synthesis, and thus the one that you need to understand the best, is the production of malonyl-CoA from acetyl-CoA, which we show here. So a molecule of acetyl-CoA is converted to a molecule of malonyl-CoA, consuming a molecule of CO2 and a molecule of ATP. This reaction is catalyzed by an enzyme called acetyl-CoA carboxylase, or ACC. Now, ACC is a carboxylase. Remember what I told you earlier, that all carboxylases require biotin as a cofactor, and ACC is no exception. ACC exists in both active and inactive forms, and the conversion of inactive to active is stimulated by citrate. Remember the citrate that comes from the citrate shuttle. And it's inhibited by malonyl-CoA, which is its downstream product, and palmityl-CoA, which is a further downstream product, and this is an example of negative feedback inhibition. The phosphorylated ACC is inactive. Phosphorylation, as in all situations, is stimulated by glucagon, and dephosphorylation is stimulated by insulin. And this makes sense because in the fasted state, you want to consume fat, not produce it. And so you want to inactivate fatty acid synthesis. In the fed state, where insulin is high, you want to activate fatty acid synthesis, not consume fats. All of the steps required to join 8 2-carbon units together to form palmitate are catalyzed by a homodimer of a large multifunctional enzyme called fatty acid synthase, or FAS. The growing fatty acyl chain is bound via a sulfhydryl bond to a cysteine residue of FAS. Initially, this is a molecule of acetyl-CoA. The incoming 2-carbon unit in the form of malonyl-CoA is bound via another sulfhydryl group to phosphopanethine, which is a prosthetic group of Fas. So the process by which fatty acids are produced are described in this picture. You can see here that we have the sulfhydryl group of cysteine, to which the acetyl group is attached. And then you have the sulfhydryl group of phosphopanethine, to which the incoming malonyl group is attached. Now, the last carbon, the carboxylic acid carbon of this malonyl group, does a nucleophilic attack of the carbon attached to the sulfhydryl group of the acetyl group. What happens then is you form a bond between this carbon, the last carbon of the malonyl-CoA group, and you've transferred the acetyl group onto that to now form a 4-carbon molecule. Now notice, though, that there are 2 carbonyl groups here, one from the malonyl group and one from the acetyl group. And remember that in a fatty acid, we only want one of those at the end. And so what we have to do is we have to get rid of this carbonyl group, and we do that by a number of different reduction steps. And those are demonstrated here. It's not important for you to understand the details of this. Just recognize that NADPH is required for this reduction. Now, at the end of this whole process, what you're left with is essentially a 4-carbon fatty acid. What happens next is this 4-carbon fatty acid gets transferred to the sulfhydryl group on the cysteine residue. Another molecule of malonyl-CoA comes in, and this process repeats itself. This time, the end result being a 6-carbon fatty acid. The next time being an 8-carbon fatty acid, and then 10, 12, 14, and 16, until finally you've produced a molecule of palmitate. So we repeat that cycle 7 times to produce the 16-carbon palmitate. The overall stoichiometry for the synthesis of palmitate by Fas is this: 1 acetyl-CoA plus 7 malonyl-CoAs plus 14 NADPHs gives you 1 palmitate plus 14 NADP pluses. Here's a summary of fatty acid synthesis. The inputs of fatty acid synthesis are acetyl-CoA, ATP, and NADPH. The output is palmitate. The key enzymes are acetyl-CoA carboxylase, or ACC, which is regulated by citrate, malonyl-CoA, palmityl-CoA, insulin, and glucagon, and fatty acid synthase. There are no associated diseases that we'll talk about with fatty acid synthesis. Now, we don't just use palmitate in our bodies. We also use other fatty acids. The fatty acid synthase, though, pretty much only produces palmitate. And so sometimes we have to make longer fatty acids. And there are enzymes in the mitochondria and the endoplasmic reticulum which can extend the length of palmitate up to 20 carbons in increments of 2 carbons each. So these molecules will convert a molecule of palmitate, C16, to a C18 fatty acid, and then subsequently to a C20 fatty acid. Now, the other thing that we need to do is we need to be able to desaturate some of these fatty acids. Desaturated fatty acids play a crucial role in cells, especially in membranes. And so you need to be able to create double bonds. Now, mammalian cells have enzymes that can create 2 different double bonds, one between the 9 and 10 position, and one between the 6 and 7 position. Now, there are, in addition to that, certain fatty acids which are termed essential. And they're essential because they have to be obtained in our diet. These are linoleic acid and linolenic acid, 18,2 and 18,3. Now, the reason we have to obtain these in our diet is because our cells cannot make them. Because our cells cannot make double bonds beyond carbon 9, as we just described. Now, these fatty acids are important because they serve as precursors for eicosanoids. Eicosanoids are a category of 20-carbon fatty acids that have really important physiologic properties. The prototype of this is arachidonic acid, which is shown below, a 20-carbon fatty acid with 4 double bonds. The important eicosanoids include things like prostaglandins, prostacyclin, thromboxane, and leukotrienes. These are paracrine hormones that produce a lot of different functions in the cell, including regulation of inflammation, regulation of blood flow, the control of ion transport, and the modulation of platelet aggregation and the clotting of blood. The first step in the formation of prostaglandins, prostacyclin, and thromboxane is catalyzed by an enzyme called cyclooxygenase. This is important because cyclooxygenase is a target for anti-inflammatory drugs, such as aspirin and other non-steroidal anti-inflammatory drugs. Here's a schematic diagram of the activity of cyclooxygenase. Here you have arachidonic acid, and arachidonic acid is converted to a prostaglandin called PGG2 by cyclooxygenase. it further acts to form PGH2, which is the precursor then for all of the prostaglandins. The cyclooxygenase enzyme is inhibited in a permanent fashion by oxygen. There is a serine molecule, or serine amino acid in a cyclooxygenase, which has this hydroxyl group, and aspirin permanently acetylates and inactivates cyclooxygenase at this point. Now, the leukotrienes are also produced from arachidonic acid. The first step in leukotriene formation is catalyzed by lipoxygenase. And this enzyme is targeted by xyluten, which is an anti-asthma medication. The reason that that works so well is that leukotrienes are important mediators of inflammation. So we've talked about how we make fatty acids, how we modify fatty acids, and how we use fatty acids in some important physiologic processes in our cells. How do we utilize fatty acids to generate energy? Well, that's done in the process of fatty acid oxidation. Fatty acids are converted back to acetyl-CoA in the mitochondria in a process that's known as beta oxidation. And this pathway involves three major steps. First of all, you have to take a free fatty acid and activate it by converting it to a molecule of fatty acyl-CoA. Then those fatty acyl-CoAs have to be transported into the mitochondria where they can be oxidized. Fatty acids longer than 12 carbons long have to be converted to their acyl-CoA form in order to be transported and oxidized. Fatty acids that are less than 12 carbons long can pass directly into the mitochondria through the membranes, and they're activated in the mitochondrial matrix. Now, fatty acyl-CoAs themselves cannot cross the inner mitochondrial membrane. In order to get across the mitochondrial membrane, they have to be modified by a molecule of carnitine, which is shown here. It's a modified amino acid. Carnitine combines with the fatty acyl-CoAs, in this case palmitoyl-CoA, to form palmitoyl-carnitine, where the palmitic acid is attached covalently to this molecule of carnitine with the loss of CoA. The acyl-carnitine is then transported across the inner mitochondrial membrane in exchange for a molecule of free carnitine. Inside the matrix, the acyl-carnitine is converted back to fatty acyl-CoA by an enzyme known as carnitine palmitoyl transferase 2. So here's a schematic diagram of this. You have a long-chain acyl-CoA combines with a molecule of carnitine, and this is catalyzed by carnitine palmitoyl transferase 1, or CPT1. That long-chain acyl-carnitine then crosses the outer mitochondrial membrane, is transported across the inner mitochondrial membrane by a specific translocase, which also shuttles carnitine back into the inner membrane space. Once in the mitochondrial matrix, then, this long-chain acyl-carnitine can be converted back to long-chain acyl-CoA by carnitine palmitoyl transferase 2, or CPT2. Now, once it's in the mitochondrial matrix, then, it can be oxidized. And oxidation is basically the reverse of fatty acid synthesis, in which acetyl-CoA groups are removed one at a time from a long-chain acyl-CoA. There are 4 enzymatic reactions required for each oxidation cycle. And it's not important to memorize the details of these. Just know that in the process, a molecule of FADH2 and NADH are produced in addition to the acetyl-CoA. Now, the complexes responsible for these 4 enzymatic reactions are different depending on the size of the fatty acid that's being oxidized. For long-chain fatty acids greater than 12 carbons, there's a long-chain complex. For medium-chain fatty acids from 6 to 12 carbons, there's a medium-chain complex. And for short chains, there's a short-chain complex for fatty acids less than 6 carbons long. Now, here's the process. You start with a long-chain fatty acid, and this is progressively oxidized until now you have 2 carbonyl groups. Then the bond between these is broken, producing a molecule of acetyl-CoA and a fatty acyl-CoA that is 2 carbons shorter than it was before. Now, knowing all this information, we can calculate then the total number of ATP molecules that are generated from oxidation of one palmitate. Now, remember that activation of the free fatty acid to the fatty acyl-CoA requires 2 ATPs, so we have to subtract 2 ATPs from the top. Now, in the process of oxidation, a cycle which turns 7 times, we produce 7 FADH2s, and we produce 7 NADHs. Those are consumed in oxidative phosphorylation, and the FADH2s produce 2 ATPs each for a total of 14 ATPs, and NADHs produce 3 ATPs each for a total of 21 ATPs. Now the acetyl-CoA that's produced from this then can enter the TCA cycle, and that can produce energy as well. For every turn of the TCA cycle, starting with a molecule of acetyl-CoA, you produce 12 ATPs. So 12 times 8 acetyl-CoAs is a total of 96 ATPs. So if you add all of this up and subtract the 2 ATPs required for activation, the total number of ATPs produced by oxidizing a molecule of palmitic acid is 129 ATPs. Now there are a couple of special situations that we need to discuss. What do we do with unsaturated fatty acids? Well, unsaturated fatty acids, when they enter fatty acid oxidation, are first treated by resaturating the individual double bonds. And there are 2 additional enzymes after the first step of the oxidation cycle which cause that to happen. And so as it proceeds through fatty acid oxidation, when it reaches a double bond, these 2 additional enzymes resaturate the double bond, and then oxidation continues to proceed. The other thing that we can deal with is odd-chain fatty acids. Not all fatty acids come in multiples of 2, especially fatty acids that are found in plants may have odd numbers of carbons in them. So what happens in this special situation is that these odd-chain fatty acids are oxidized until the last step, which produces a molecule of propanil-CoA, a 3-carbon fatty acid, essentially. And this is oxidized via a process we'll discuss later in amino acid metabolism. The regulation of fatty acid oxidation occurs via 2 processes. One is simply the availability of substrates and cofactors. If the NAD+ levels and FAD concentrations are high, the reactions tend to proceed more rapidly. On the other hand, fatty acid oxidation can also be regulated by malonyl-CoA. Now you'll remember that malonyl-CoA is an intermediate in the fatty acid synthesis. What malonyl-CoA does is it inhibits CPT1, the rate-limiting step in fatty acid oxidation. So what this does is it ensures that fatty acid oxidation and fatty acid synthesis are not occurring at the same time. That's a problem because that would create a futile cycle in which we're producing fatty acids and then immediately degrading them back to acetyl-CoA. Now, one of the things that that we learned about acetyl-CoA before was that acetyl-CoA is not a substrate for gluconeogenesis. Remember that pyruvate dehydrogenase, the process by which we convert pyruvate to acetyl-CoA, is not a reversible reaction. So acetyl-CoA cannot be used to make more glucose. So the only things that we can do with acetyl-CoA then are basically 2 things. One, we can oxidize them in the TCA cycle. Number 2, we can convert them to ketone bodies. And the path that's taken depends upon the energetic balance of the cell and the organism at the time. The major organ for producing ketone bodies is the liver. So under conditions of low blood glucose, much of the oxaloacetate is shunted to gluconeogenesis, which has been activated by glucagon. Consequently, the TCA cycle is relatively inactive because a lot of its intermediates have been shunted away to gluconeogenesis. And so what can happen in this case then is that the acetyl-CoA, rather than entering the inactive TCA cycle, is converted to ketone bodies, acetoacetate and beta-hydroxybutyrate. These ketone bodies are especially important for the brain. The fatty acids cannot be transported across the blood-brain barrier. Consequently, the brain is almost entirely reliant upon glucose for energy. So in the fasted state, when glucose levels are low, then the brain needs some other source of energy, and they can get that source from ketone bodies. Therefore, the activation of ketogenesis during hypoglycemia is really important for the maintenance of nervous system function. Ketone bodies can also be used by other tissues such as muscle, but the predominant role is in providing energy to the brain. Now, there are defects in fatty acid oxidation as illustrated by the following clinical vignette. A 5-month-old girl is brought to the emergency room because her parents had difficulty arousing her from sleep. Once she woke up, she vomited, but she remained very lethargic. She's just recently begun sleeping through the night, and they've noticed that she awakes more lethargic the longer she sleeps. Usually once they feed her, though, she behaves normally. Pregnancy and birth were normal. Growth and development have been normal. It's significant that the first child of these parents died of sudden infant death syndrome. You do an exam and you notice that the patient's lethargic and difficult to arouse. Vital signs are normal. Physical exam is unremarkable except for moderate hepatomegaly, and there's no other stigmata or signs of liver disease. You immediately order a capillary blood glucose, which comes back markedly decreased at 15 milligrams per deciliter. And the urine dip is negative for ketones. Now, why is that interesting? Well, we'll talk about that in just a minute. So what disease does this patient have? This patient has medium-chain acyl-CoA dehydrogenase, or MCAD, deficiency. MCAD catalyzes the first step in oxidation for fatty acids of approximately 6 to 12 carbons in length, the medium-chain fatty acids. When it's deficient, the patients can't mobilize fatty acids for energy during periods of relative starvation. So the symptoms tend to appear as infants begin to sleep for longer periods of time at night as they take more time in between feedings. With the severely decreased fatty acid oxidation, the tissues are more reliant upon glucose for energy, and a profound hypoglycemia results. This is particularly true for the central nervous system, which relies upon glucose for energy. Normally, the hypoglycemia would be accompanied by ketosis, and this would help the brain continue to function even in the face of hypoglycemia. But without fatty acid oxidation, the acetyl-CoA necessary for producing ketone bodies can't be produced. And so what you end up with then is a hypoketotic hypoglycemia. The signs and symptoms of MCAD deficiency include hypoglycemia, vomiting, lethargy, encephalopathy, seizures, cardiopulmonary arrest, and sudden death. Hepatomegaly results from the accumulation of medium-chain fatty acids in the liver. Now, homozygous MCAD deficiency has been linked to a certain fraction of sudden infant death syndrome cases, similar to the type of thing that is described in this case. You treat this disease by giving frequent high-carbohydrate meals and giving carnitine to increase fatty acid oxidation. The long-term complications include neurologic, developmental, and behavioral problems, and these are seen even in children who are treated. So here's a summary of fatty acid oxidation. The inputs are palmitic acid or some other fatty acid. The outputs are acetyl-CoA, NADH, and FADH2. The key enzyme is carnitine palmitoyltransferase 1, or CPT1, which is regulated by malonyl-CoA. Associated diseases with this include MCAD deficiency. This concludes